speaker, Sebastian Bobadilla-Suarez, who is a postdoc in the Department of Experimental Psychology at UCL, working with Prof. Brad Love, and he will be presenting us his work on the neural similarity in bold responses and multi-unit recordings.
Cool, thank you. Can you see that? Okay, I'll start my stopwatch. Thank you everyone for coming to see my talk on neural similarity in fMRI and multi-unit recordings. Thanks to the organizers of Neuromatch and Arianna and Valentina for hosting us. So I'll present work in collaboration. Well, my name is Sebastian Bobadilla-Suarez. I'll present work with Brad Love. I'm in his lab at UCL, and a master student, Marilena Lemonari, and supported by Wellcome Trust, Royal Society, and NIH. It's also in collaboration with Earl Miller's group at MIT. Well, Marcus Siegel's at Tübingen now, but he was at MIT when the work was done. So yeah, to get started, well, how does the brain represent similarity? That's what I'm interested in. So just to give an example, there's a person in a scanner, you show them the picture of a robin, right? So you get a brain activation, and that's the representation that we can work with. You can do the same for a sparrow, right? So you can compare the brain activations as opposed to the images themselves to see how the representation differs for each one of these birds. And then compare that to similarity with the robin and the parakeet, for example. So then we have sets of similarities, right? And then we're interested in quantifying these similarities at the brain activation level. So how do we pick these similarity measures? Because each measure has its own assumptions, right? It'll focus on different aspects of the data, of the brain data. The way we pick the measure, the one that best describes the brain data, well, it'll reveal something very fundamental about the neural code in a way, you know? Each measure, like I said, has hidden assumptions that has mechanistic and theoretical implications of how the brain is representing stimuli. So the first part of this talk is fMRI in humans. And in that study, it's published already in Computational Brain and Behavior. We focus on a set of measures that we're interested in. Each family of measures focus on different summary statistics of the fMRI data. So some will focus more on like an angle type information, or maybe the magnitude of the numerical vector. So we're picking numerical vectors here for voxels, or maybe more distributional information. So just to give a quick example, like this is a similarity measure like Euclidean distance would say that objects A and C would be more similar here. Whereas a measure that focuses on angle would say that A and B are more similar in this two-dimensional voxel space. A common measure that people use in this type of studies is Pearson, for example. But I think we have to question the choice of measure to really tell us something about how the brain represents stuff. So like I said, this is published work. The work actually focuses on two previously published fMRI data sets, one from Mike Mack and Ali Preston, the other one from Optabeek and Stephanie Bracci. So it's two different stimulus sets that we're interested in. One that just, in their task, they just show geometric shapes in this data set here. And in the other one, just natural images. So it's different stimulus sets. And we're interested to evaluate the measure in each of these data sets to see if there's differences between the data sets, but also between brain regions. Since we're interested in these differences between brain regions, you know, we can't really just rely on behavior because different brain regions might have different measures that they like to work with, or depending on the task as well. So it goes well with what Maria was talking about in her talk about this interaction between the stimulus and the task demands. So for this methodology, the logic is that similar stimuli are more easily confused, very intuitive way of thinking about things. So we can use a classifier to decode these brain activations to differentiate between a robin and a sparrow, for example. And we can use the confusion matrix of the classifier as our reference, as the gold standard of similarities. We can compare that to the other similarity matrices that we're interested in, like Pearson or Mahalanobis. And we constrain this to areas that have good decoding accuracy, so that actually have enough information for us to make this comparison. And just to keep it short, I'll just say that in the top row, we have these geometric shape data. So this is more of a visualization, but in blue is the Euclidean measure, and in red is the Mahalanobis measure. And just in general, yeah, we see that Euclidean type measures and magnitude type measures work well with the geometric shapes data set, whereas on the bottom row, Mahalanobis totally dominates in the natural images data set. So if you want more information, you can look at the paper on that. Just to pivot a bit here, this is really exciting work that we're doing with Errol Miller's data on multi-unit recordings in macaques. And what does this bring us compared to what I just showed you? Well, here we can look at more temporal resolution, right? So look at the temporal statistics of the neural activation and see what we can find there with the same kind of logic that I was talking about. So it's a hotly debated subject, whether it's rate measures or temporal-based measures for spike trains that are important. But yeah, like I said, at least we can do this comparison here, and you can't really do it with slow fMRI data. So it's based on this paper in Science from Errol Miller's group, if people want to take a look. So in that paper, well, just to mention, the data has the advantage that, just like Maria was talking about, the task demands will emphasize different dimensions of the stimulus space. And that's something we're really interested in as well, just because that basically also tells you something about the representation and the similarity between the stimulus, what dimensions are being emphasized in the representation. So the monkeys, Rex and Paula, and that's not them, but anyways, they did thousands and thousands of trials discriminating random dot motion. But before they see the random dots, they would see a cue to tell them if they have to discriminate the motion or the color. It was 100% coherence for the dots. And we can see in panel C here, that's the actual representation of the stimulus space. So you have color on the y-axis and motion on the x-axis. And this is obviously, it was defined by the experimenters, so hand-coded in a way. And then, yeah, you give a different response, left or right, depending on if it's motion or color that you're discriminating. Well, Rex and Paula in this case. And it's a massive data set. It's really nice data set that they looked at over, did like 6 areas in the brain, sometimes simultaneously over many, many sessions. So really cool stuff. Like I said before, here we can actually look at the spike trains, right? We have different neurons with different spikes, different time points, and we have these different areas to work with. So just at a broad scale, we can see, okay, is timing information relevant, or is the average firing rate what's relevant, as people normally talk about? So what summary statistics should we consider? We chose 3 measures for each class of measure. On the left-hand side, we have ISI, which is about the interspike interval, spike and sync, which I won't really go into too much detail, but I'll give you the reference. And then the ones we talked about before, Euclidean, Pearson, Mahalanobis, defined on a certain area, of brain region or lobe. So that's the reference for the timing measures that we took into consideration. So yeah, just to remind you, this is the stimulus space that the monkeys were working with. So each stimulus here in this graph was basically represents the random dots that they saw at some point. And they would have to give a left or right response depending on what they needed to discriminate, color or motion. Just to make, again, the long story short, I just want to clarify here that the logic is a bit different in that before we were using a confusion matrix of a classifier to infer like the gold standard similarities to compare with. In this case, we're using the ones defined by the experimenter. So we have these stimulus coordinates that I just showed you back here, right? These are defined by the experimenter, and we can use that to get our similarities as our gold standard, if you will, and then use that to compare all the other measures that we see in the empirical data. And what we see is actually that this ISI measure does relatively well, quite well compared to the other ones. In general, the timing ones seem to do better than the firing rate ones, but ISI just really has good performance. Something else that we were interested in, it goes well with what Maria was saying, is that the task demand, so whether you're discriminating color or motion, it'll change the representation. We've seen this in human data, for example, or in monkey data. And just from the behavior, for example, you see that the representation stretches along the relevant dimension. So if you're discriminating color, it'll stretch the y-axis. Complementarily, if the monkeys have to discriminate motion, you'd expect the stretching along the motion axis. This helps the monkey solve the task because it makes the discrimination easier if they represent things this way. And this has been shown also in fMRI. I'm not sure if you can see that reference, maybe it's cut off. But anyways, by Mac, Love, and Preston and PNAS, if people want to check it out. So with humans, they had a category learning model that would look at these emphasis on the relevant dimension depending on the type of problem. So you would get these attentional markers of the stimulus representation, and that would be tracked in hippocampus, for example, in that paper if people want to take a look. But again, the stimulus was hand-coded by the experimenter. So you're kind of already guessing what the representation is like, which is why we tried to actually guess it with a classifier in the first part of the talk. So yeah, that's just what I said right now. So how to get at, oh, sorry. Anyways, I don't know if it's cut off at the bottom, but basically to get around this issue of hand-coded representations, another strategy is to try to infer them with a model that learns to do the task as well. So in this case, we can use a deep learning model. So this model, it was done by Marilena Lemonari, a master's student. It doesn't have access at all to the neural data. So all it has access to is the same picture that the monkey saw to actually do the task. So the pixels and the label of the stimulus, depending on if they had to discriminate the motion or the color. So it basically has to learn what the monkeys were doing. We basically got a CNN that was pre-trained, VGG16, fine-tuned it to the images that the monkeys saw to discriminate the images. And then we got the penultimate layer of activations, and we fed that into an LSTM. So it's another type of learning model that's good for sequence learning. And basically, yeah, the model learned to do the task. It basically learns to do cognitive control, provided only with that supervision signal, the stimulus labels, and a pre-trained visual system, which, the monkeys also have a pre-trained visual system. So yeah, I thought that was pretty cool. The model also stretches the dimensions along the relevant, dimension, depending on what they have to discriminate. So just take-home messages. So similarity measures can differ across tasks, but in the fMRI, we see that they stay constant across regions. And they can benefit from timing information. That's what we see in the multi-unit recordings. And they can also, across these different modalities, the model, the LSTM model, fMRI, multi-unit, they can provide a window on the stimulus dimensions, on the stretching, depending on the task demand. And in this case, it's a cognitive control process based on task demands. You get these attentional effects. And yeah, I'll just give my contact information, and thank you to my collaborators, to everyone who organized and our funding. And yeah, if anyone wants to contact me about anything, I'm more than happy to chat.
Thank you. Thank you very much for presenting this extremely interesting method that, as you said, has a lot of connection with what we heard before, and it definitely can be applied. And I already see a question that Benjamin Silver is asking, so I'm going to read it for you. Has anyone ever compared similarity data from a classifier to self-reported similarity data? So for example, a subject that rates a parakeet on a scale from a robin to a sparrow, and investigating what might cause varying levels of divergence between the two,
So yeah, I don't know off the top of my head, but yeah, definitely people have looked at behavior. That was what was normal before. As far as using a classifier on a region, I actually think that the fMRI data set we were basing on from Optabeak Lab and Stefania Bracci, they actually look at something similar to this. I don't know if that answers the question, but yeah, definitely. Yeah. I don't think anyone has actually tried to focus on just inferring it directly from the neural activity itself. Because behavior will only tell you something about that final stage, that output stage. But different brain regions might act differently, although that's not what we see, right? in their data.
Right. We have another question. Yeah, and we have time. Jules Burchard is asking, would dimension stretching relate to the dynamic range adaptation and efficient coding?
Oh, man. Dynamic range of adaptation, efficient coding. I'm not sure. Sorry, I'm not sure I understand that question. So are they saying that there might be like a cap on the stretching? I'm not sure.
Or at least a relation between how much a given dimension would be stretching for a given subject and how much that subject is.
I'm not sure how to answer that question, because stretching is obviously a relative term to the other dimension. I don't know how you can measure it like in an absolute sense, the stretching, because it's always the ratio between the scale of dimension Y versus dimension X. So it's the problem you always have with attention, is that one dimension is being inhibited or is that the other dimension is being tuned, and you got to gain response gain on this other dimension? So I'm not sure how you would answer that, but I have to think about it. Yeah, thanks.
Right. Yeah, great question, because it's triggering a lot of reflections in our audience. So thank you very much for your talk.
Thank you.
Yeah, please feel free to reach out to him online. And
cheers.
we can move to our last speaker.